Hello students, welcome to the channel Physiology Redefined. Now is about the factors affecting erythropoiesis. In my earlier video I explained what is erythropoiesis and how the erythropoiesis takes place. There I told that erythropoiesis is the process of mechanism of formation of the RBC, matured RBC from the bone marrow, from the pluripotential stem cells or otherwise known as hemocytoblast. Now in this video, I will discuss about what are the factors that affect the erythropoiesis. What are the factors that influence the erythropoiesis, that can alter the rate of erythropoiesis? So, factors affecting erythropoiesis. Factors affecting erythropoiesis. So, to know about the details of the factors which influence the erythropoiesis, please keep watching till the end. And if you like this class, don't forget to share it to your friends and subscribe the channel if it is not yet subscribed. So let us see what are the factors that affect the erythropoiesis. So there are three categories of the factors. First one is known as the general factor. First factor is known as the general factors which influence the erythropoiesis, rate of erythropoiesis. Second factor is known as the maturation factor. Maturation factor. And third one is known as the hormonal factor or hormonal or growth factors. Growth factors. So there are three categories of the factors which can influence, which can alter the rate of erythropoiesis. So first I will talk about the general factors, second I will talk about the maturation factors, and third I will talk about the hormonal or the growth factors. So let us see what are the general factors. General factors includes two types of components. One is known as the hypoxia and second one is known as the dietary factor. Dietary factor. So first, what is hypoxia? Hypoxia is the lack of oxygen to the tissue. Deficiency of the oxygen to the tissue. That is the hypoxia. So in response to hypoxia, or hypoxia is the very potent stimulus, important stimulus which can increase the erythropoiesis, rate of erythropoiesis. How the hypoxia, lack of oxygen in the tissue, increases the erythropoiesis? Here you see. So hypoxia or oxygen lack, this results the stimulation and release of some hormones or some factors which are released by the kidney, and that factor is known as the renal erythropoietic factor. Renal erythropoietic factor or otherwise known as erythrogenin. Now this erythropoietic factor, renal erythropoietic factor, which ultimately is responsible for conversion of the plasma inactive erythropoietinogen to form the erythropoietin. That means erythropoietin is mainly released in response to the factor which is from renal source. That is known as renal erythropoietic factor. Now, kidney, as I told, kidney has got main role to release ultimately the erythropoietin. And which cells of the kidney? This kidney has got the interstitial cells very close to the peritubular capillary plexus. Very close to the peritubular capillary plexus, there are interstitial cells. And these interstitial cells, so this is release of renal erythropoietic factor. Renal erythropoietic factor or renal erythropoietic REF, and this is released by mainly the interstitial cells close to the peritubular capillary plexus of kidney, and partly by Jaypee cells of juxtaglomerular apparatus. So source of this erythropoietic factor, this is the renal source. This is the renal source. This is the renal source, which is around 85%. So 85% of the erythropoietin is released under the influence of the kidney or from the kidney under the influence of renal erythropoietic factor. So 85% source of erythropoietin is of renal source. Remaining 15% source is the extra-renal source. Extra-renal source, which is around 15%, and this is done by liver and the cells of tissue macrophage system. Tissue macrophage system. So these cells of tissue macrophage system and the liver are the another source of the erythropoietin, which collectively are responsible for increasing the rate of erythropoiesis. How? Here you see. So as I told, release of renal erythropoietic factor REF, and this erythropoietic factor is responsible for conversion of erythropoietinogen to form the erythropoietin or in short EP. So erythropoietin is now formed under the influence of renal erythropoietic factor, mainly released from the kidney. And once the erythropoietin are released, then this erythropoietin has got diverse action to increase the erythropoiesis. So this erythropoietin increases Rbc formation or erythropoiesis, and this Rbc formation is influenced by erythropoietin by diverse mode of activity. It has got diverse mode of action. How? First one, it stimulates the bone marrow. It stimulates the multipotential uncommitted stem cells, pluripotential uncommitted stem cells to be differentiated into committed stem cells. So it chiefly acts on the bone marrow, stimulates the pluripotential stem cell to be differentiated quickly into the committed stem cells. Secondly, this is done, Rbc formation is done by stimulating the bone marrow. Secondly, it also stimulates the all phases of the RBC formations. That means committed stem cells to the mature RBC. All the stages, differentiation of the RBC up to mature RBC through all the stages are also enhanced by the erythropoietin. Third action is that it also increases the globin synthesis and ferritin synthesis. Globin synthesis and ferritin synthesis, and thereby responsible for increasing the globin formation and heme formations. So, erythropoietin has got triple action. Number one is that stimulates the hemocytoblast cells, pluripotential stem cells to form the committed stem cells. Second one, it stimulates the committed stem cells to be further differentiated into mature RBCs. And thirdly, thus it increases the globin formation, it increases the ferritin formation, and thereby increases the hemoglobin formation that is responsible for oxygen carried via the RBC. So this is how the hypoxia influences or increases the RBC formation. Second factor is known as the dietary factor. So dietary factor includes different minerals. What are the minerals? Chiefly, there is the iron, there is the copper, there is cobalt, and there is nickel. So these are the chief minerals which are responsible for increasing the electropoiesis, increasing the rate of erythropoiesis, and these are available in the diet and thereby included in within the dietary factor. So iron, as you know that iron is responsible for heme synthesis. This is responsible for heme synthesis and thereby increase the RBC formation. Copper, this increases the iron absorption from the diet to the Jaypee and thereby increases the RBC formation. Cobalt and the nickel, these are responsible for increases utilization of iron to synthesize hemoglobin and thereby increases the RBC formation. So these are the different minerals which are one of the prime important dietary factor. Second one is the protein. Second dietary factor is the protein. As you know that hemoglobin, RBC contains the hemoglobin. Hemoglobin is comprised of heme and the globin, and globin is basically the proteins. These are formed from the amino acids. So protein as a diet is very, very important for formation of the globin and thereby the hemoglobin and thereby RBC formation. So proteins, these are responsible for formation of the increased globin synthesis and thereby these are responsible for increasing RBC formation or erythropoiesis. Third dietary factors are the vitamins, different types of vitamins. So what are those vitamins? First one is the vitamin C. Second one is the vitamin B12. And third one is the folic acid. folic acid. Vitamin C, this is responsible for heme synthesis, globin synthesis, and thereby increases the RBC formation. Whereas vitamin B12 and the folic acids, they help in maturation of the RBC, and thereby they are included under the maturation factor. So I will talk about details of the vitamin B12 and the folic acid under this category. That is the second factor, which is known as maturation factor. So this is all about the general factors. So let us see what are the maturation factors. So as I told, or from the name it implies that maturation factors are the factors which helps in maturation of the RBC. So what are those factors? Under these, there are two categories of the factors. First one is known as the extrinsic factor, and second one is known as the intrinsic factor, which was first identified by scientist Cassel and thereby known as Cassel's intrinsic factor. So extrinsic means outside from the body. What are those substances? Vitamin B12 and folic acid. These two substances, vitamin B12 and the folic acids, these are responsible for DNA synthesis. And DNA is responsible for maturations of the RBCs. Or RBCs are getting matured, are getting differentiated because of the influence from the DNA. And thereby, if the vitamin B12 and the folic acids are absent in the diet, what's happened? The RBC will not be matured. In the blood vessels, there is a large size immature RBCs will be there, and those are known as the megaloblastic cells. And such a conditions, the person will suffer from anemia, and that anemia is known as the megaloblastic anemia. So deficiency of these things, deficiency of this one results the large size RBC, immature RBC, and thereby the functions of the RBC will be lost. Oxygen carriage will not take place properly, and thereby the person will suffer from anemia, and such anemia is known as the megaloblastic anemia or maturation failure anemia or megaloblastic anemia. So both are because of the vitamin B12 and the folic acids, which are responsible for maturation of the RBC within the differentiation process. And second one is known as the Cassel's intrinsic factor. So what is Cassel's intrinsic factor? You know that within the stomach there is a parietal cells or oxygenetic cells, which is responsible for secretions of the HCl, hydrochloric acid. Now, along with this HCl secretion, these parietal cells or the oxygenetic cell has got another action. They synthesize another very important substances, which is known as the intrinsic factor, and that factor is known as Cassel's intrinsic factor. So what does it do? Cassel's intrinsic factor, they binds the vitamin B12, dietary vitamin B12 and the vitamin B12 especially in the stomach. So once vitamin B12 enters into the stomach, it should be protected from the oxidizing agent of like hydrochloric acid. Hydrochloric acid as a strong oxidizing agent can destroy the vitamin B12. So, vitamin B12 from the diet has to be protected. Who will protect? This caseal intrinsic factor. So, caseal intrinsic factor binds the vitamin B12. It binds the vitamin B12 and prevent the hydrolysis or oxidation of the vitamin B12 from the hydrochloric acid action. So hydrochloric acid cannot hydrolyze them, vitamin B12, and thereby vitamin B12 from the diets are protected in the stomach. And this caseal intrinsic factor, vitamin B12, this caseal intrinsic factor binds with vitamin B12 in stomach. This is secreted by parietal cells of stomach. Parietal cells of stomach. So which binds with vitamin B12 in stomach, protect them, and they carry the vitamin B12 up to the level of ileum, which is the chief site from where the vitamin B12 is absorbed by the gastrointestinal tract. So this protect them, brings to ileum, and thereby these are responsible for increased vitamin B12 absorption, and thereby these are responsible for maturation of the RBC, maturation of RBC. So RBC production will be increased. So again, deficiency of this caseal intrinsic factor, that happens in case of gastric atrophy, atrophy of the gastric mucosa. Atrophy of the gastric mucosa, in such a case, not only the HCl secretion is suppressed, but also caseal intrinsic factor secretion is suppressed, is decreased. As a result, it cannot bind with vitamin B12. So vitamin B12 is not protected. As a result, vitamin B12 will be hydrolyzed under the actions of the HCl, whatever HCls are there, under the action of those HCl, vitamin B12 will be hydrolyzed. So it will not be protected. So ileum will not get any vitamin B12 to absorb the vitamin B12 into the blood, and thereby no maturation of the RBC will take place, and thereby again megaloblastic anemia will take place. And such megaloblastic anemia, in case of such megaloblastic anemia due to the deficiency of the caseal intrinsic factor, due to the deficiency of this caseal intrinsic factor, is known as the pernicious anemia. Pernicious anemia. So deficiency of the vitamin B12 or the folic acid, that results the megaloblastic anemia. Deficiency of the caseal intrinsic factor also results the megaloblastic anemia. Both are responsible for maturation failure anemia. But if the maturation failure anemia or if the megaloblastic anemia is resulted due to deficiency of caseal intrinsic factor, then it is known as pernicious anemia. So this vitamin B12, folic acid and the caseal intrinsic factor, they are responsible for increasing the RBC formation, and this is why all these substances are responsible for increasing RBC formation, and thereby all these things are collectively known as hematinic principle. Hematinic principle. So this hematinic principle means these are vitamin B12, vitamin B12 plus folic acid plus Cassell's intrinsic factor, all are collectively known as hematinic principle, which are responsible for RBC formation, So this is all about the maturation factors. Second category of the factors that influence the erythropoiesis. Now is about the hormonal factors or the growth factors. So there are different types of hormonal factors or the growth factors. First one, as I talked about the erythropoietin, which I already talked. So erythropoietin increases the RBC formation, and as the erythropoietin is the hormone, it can also be included under the hormonal factors. Second one is the androgen. That is testosterone mainly. So testosterone stimulates the bone marrow and stimulates the pluripotential stem cells to form into committed stem cells and also other stages of the RBC formations. So this androgen or like testosterone increases, this stimulates bone marrow, and thereby there is a increased RBC formation. And this is why in case of male the RBC count is more. Why? Because of this factor, because male has got testosterone, that is the androgen, which stimulates bone marrow to stimulate the pluripotential stem cells, which is differentiated to the mature RBC, and thereby RBC formation is increased. Second one is the adrenocortical hormones. Third one is the thyroid hormones. Fourth one is the growth hormone. So all these hormones, adrenocortical hormones, thyroid hormones, growth hormones, they increases the hemoglobin synthesis. And thereby increase RBC formation. Mainly these are responsible for increasing RBC formation by influencing the hemoglobin synthesis. Another hormone is known as the estrogen. This estrogen is negatively associated with, so all these factors are directly related with erythropoiesis. Whereas estrogen is inversely related with erythropoiesis. That means estrogen concentration, if it is more, the erythropoiesis will be less. If estrogen content is less, erythropoiesis will be more, inversely related. In case of female. In case of female, estrogen is more. As the estrogen is there, under the influence of estrogen, erythropoiesis will be suppressed, and this erythropoiesis will be suppressed by estrogen by again inhibiting the hemoglobin synthesis. So thereby estrogen also another hormone which is negatively correlated with the rate of erythropoiesis, whereas other hormones are positively correlated with the RBC formations. Now next category of the factors, these are rather growth factors, which are known as the colony stimulating factors, CSF, colony stimulating factors, CSFs. And these colony stimulating factors, these are responsible for stimulation of pluripotential stem cells, that is hemocytoblast, to form the comitate stem cell. Comitate stem cell. That means the hemocytoblast is converted very quickly, rapidly, or differentiated very rapidly to form the comitate stem cells, that is the myelin stem cell series, from which the BFU and the CFU or blast forming unit and the colony forming units are formed, and thereby RBC formation is getting enhanced, rate of RBC formation is getting increased. And last one is known as interleukins. Different interleukins like interleukin 1, interleukin 3, interleukin 6 and 9. These are also responsible for RBC formation. How? These are responsible for stimulation of comitate stem cells, that is comitate stem cells, to form the mature RBC. So coronary stimulating factor is responsible for stimulation of the pluripotential stem cells to comitate stem cells, whereas interleukins are responsible for stimulation of all the phases starting from comitate stem cells to the mature RBC. And thereby these are the different factors which are responsible for regulating the erythropoiesis. So this is all about the factors influencing or factors affecting erythropoiesis. If you like this video, if you like this class, please subscribe this channel and press the like button to motivate me so that I can bring the same type of lectures for your guidance or for your help. Thank you.